Okay, and next lecture, again about light in the life. Ilya Yampolsky, Chemistry of the Light, Fluorescent Proteins, Luciferins, and Luciferase. You are sleeping?
Thank you, Sergey. Дорогие коллеги, dear friends, as a member of administration of our institute, I'd like to first, with great pleasure, welcome most warmly our guests from Russia and from abroad. And I hope your coming here this time will continue the old ones and give rise to many novel collaborations and inspiration and cooperation across the institutes, the cities, and the countries. Okay? Well, then, I'd like to make another short introduction to begin with the very beginning. I believe that scientific tradition is one of the most important features in our sort of business, and I'd like to praise the milestone achievements of Professor Sergey Lukyanov. He's the chair of our session today. This achievement inspired many of us, his scientific editors, including me, to start our own research. I mean, the discovery in 1999 of the red fluorescent proteins, which deeply influenced many fields of modern biology. Having said this, let me shift your attention to the main topic of my today's lecture. And I would like to tell you about one of our projects dedicated to answering the question first posed as early as in the 4th century before Jesus Christ by Aristotle, who reported his exploration of the so-called foxfire. Now we know that this phenomenon is caused by luminescence of fungal mycelium infecting the dead wood. As for today, we know more than 100 species of luminous mushrooms, mostly inhabiting South Asia, East Asia, but also present in North America and in Europe. For example, this one is a European species. If we shift forth from this particular problem to a more general picture, we would reveal that the phylogenetic diversity of bioluminescence is actually very high. You can see in this picture marine species, very many of them, including fish, jellyfish, sea combs, worms, microorganisms like dinoflagellates or radiolarians, terrestrial species like bacteria, soil worms, insects, mollusks, and of course the subject of our today's interest, mushrooms. How does actually work the emission of light by a living organism? In general, this is caused by oxidation of a low molecular weight substrate called luciferin by molecular oxygen under the catalysis of a specific enzyme, luciferase. And this reaction, all these three components together, give light. But the chemical diversity of bioluminescent mechanisms corresponds tightly with the phylogenetic diversity. And therefore, today's current estimation is that in nature there exists over 40 different biochemical mechanisms, meaning different luciferins and different families of luciferases. And until recently, only a handful of this substrate, the minority, has been known. Namely, you can see here the list of known luciferins, namely of bacteria, fireflies, some marine organisms, soil worms, and one mollusk. In these 3 boxes, I hope we'll say a few words about them later. Okay, to be short, I will not focus on this, I'll just show you the picture of one luciferin that we have established from the soil worm. And also our current project, yet unpublished, is dedicated to the exploration of bioluminescent chemistry of this marine organism with notorious classification, fireworm. And here we were also able to elucidate the family of complex organic molecules with their interconversions. But I will not focus on this. This is just to give you an impression of the field. Well, if we now shift back again to luminous mushrooms, you can see that the mycelium may be luminescent like in foxfire, or also the fruiting bodies. To cut the long story short, we also have been able to elucidate the structure of the key molecule in this mechanism, namely luciferin of higher mushrooms. It turned out to be unique for all species of bioluminescent mushrooms. It has an unusual mechanism of action, including the formation of Diels-Alder intermediate and the retro-Diels-Alder scission of carbon dioxide gives rise to oxyluciferin and the quantum of light. So, summarizing the few previous slides, we have been able to discover 3 novel molecules that are responsible for bioluminescence of different creatures. But then our interest naturally shifted to the proteins that are involved. And concerning the fungal case, we have been able recently to clone fungal luciferase. Here you can see the colonies of yeast emitting the light, which can be seen by naked eyes. Then we expressed this luciferase in many organisms and cells, including human cells. These are also human cells, I suppose. We demonstrated the possibility of in vivo imaging in living mice and compared our newly found luciferase with the well-known insect luciferase, and we see that their performance is comparable. This is another imaging attempt, probably proof of principle attempt of imaging of Xenopus embryo. And now, this is, I think, a culmination slide of today's talk that summarizes the mechanism of fungal bioluminescence. Actually, probably somewhat similarly to citric acid cycle. In this case, we deal with the caffeic acid cycle. And you can see that caffeic acid is first used as a substrate by the enzyme belonging to the class of polyketide synthases. And the molecule of luciferin precursor is formed. Then the luciferin precursor is transformed to luciferin itself. Then luciferin is used by luciferase to produce light, and then the product of oxidation returns back to caffeic acid. How did we find this? After cloning of fungal luciferase, we looked at the genome of several species of bioluminescent mushrooms and found that luciferase belongs to a gene cluster. And looking at these genes, we supposed that they might be involved in the biosynthesis of luciferin. Then we cloned each one of them separately and together and proved its function by heterologous expression. So what we have here is the gene of luciferase, the gene of polyketide synthase, this one, blue, and two other proteins that are responsible for the rest of the transformations. What are the possible applications of this finding? Actually, the system I presented to you today is the first eukaryotic bioluminescence system comprising not only luciferin and luciferase, but also the full pathway of luciferin biosynthesis, making the system like portable and transferable in its fully operating state into any organism of choice. And our first idea, which is now under exploration actively, is to create bioluminescent plants that would decorate the cities, probably, and gardens of rich people. Okay. You can see a number of papers published within the framework of this project. A couple of entertainment slides, like the cover pictures in different journals. Actually, this is original artwork by one of the members of our lab. These two pictures, this one and this one too. Also, an entertainment slide showing the side cover of Science Advances journal, a couple of newspapers highlighting our projects recently. I think I will not bother you anymore with complicated information, and I would like just to finally praise the big team that worked from different countries, from our institute mainly, but in several other Russian and international institutes. And thank you for your attention.
Yeah, thank you. Are you trying to get some money from green energy support funds? No, it's very
Trying to get money everywhere.
Okay. Do you have any questions, colleague?
So, yeah, thank you for this stimulating talk. But what do you think? Let's have a dream, not maybe color dream. Because you discovered the apeiron generally, the apeiron which regulates the colors. What do you think? You may change, certainly, by mutations, different part of this, or stopping by genetic engineering, and switch color, yes? Do you have this?
Yes,
and
sure.
even time of color, and so
I
it's
didn't-
maybe very, yeah, it's
Thank you for the question. I didn't tell about this branch of the project.
Yeah.
We got the branch like you're talking about. Modification of the substrates and also of the protein.
I think it's protein
parts.
in this case, yeah.
In case of enzyme and substrate, we can modify both substrate and enzyme. Yeah,
Two-dimensional screening approach.
yes, and
I guess.
we are practicing this.
Yeah, thanks.
Thank you. More
Mike wanted
questions? Oh, okay.
I have a question on the photophysics of the system. Have you any information on the thermodynamic efficiency of light production in this fashion compared, for instance, to a modern LED?
Thank you. As far as I know, LED energy efficiency is close to 1, yeah, to 100%, probably 70% or so. In case of bioluminescence, the situation is the same, basically. One molecule of substrate generally gives 1 quantum of light with a quantum efficiency of about close to 100% probability. But biosynthesis of luciferin takes much energy, actually. So if you look at the full biochemical cycle, so the efficiency would be probably about 100 times less. But still, this is affordable for the living creatures. No. You can see the mushrooms, they do glow.
Yeah.
They are just saprophytic, and they don't catch and eat other creatures. Yeah? They have enough energy to maintain this.
We will wait. Okay. Do you have any questions? Okay. Thank you, Ilya.